For decades, the mechanistic narrative of Alzheimer's disease has been framed around failure. The prevailing models describe a brain where clearance systems break down, barriers leak, and amyloid plaques accumulate unchecked. The grammar of the disease is almost entirely one of a protective apparatus giving way. But that model struggles to explain the clinical reality. The best structural correlate we have for cognitive impairment in Alzheimer's disease is not the plaque count. It is the loss of dendritic spines, the physical connection points between neurons. And this loss is not a passive consequence of toxicity or cellular wear and tear. As we can see in this rendering, the spine dismantles its own internal scaffolding from the inside out. It receives an instruction to take itself apart, and it executes that command. To understand how a synapse is ordered to destroy itself, we have to look outside the standard Alzheimer's literature. We have to look at a 27-year body of work from Carla Shatz's laboratory, which originally sought to answer a completely different question. How do healthy brain circuits refine themselves during development? That developmental research arrived at a precise mechanism. The brain actively prunes its connections using a physiological machinery built from immune system proteins. In Alzheimer's disease, that exact same developmental pruning program is running late in life, and it is running without a stopping role. The central executor of this program is a highly specific receptor sitting on the neuron surface. In mice, it is called PIR-B. In humans, it is known as LIL-RB2. This requires a shift in how we view the pathology. The mechanism eliminating these synapses contains no faulty parts. The synaptic break is in perfect working condition. It is simply being pressed by the wrong hand. To understand the disease state, we have to establish what this receptor is doing in a healthy brain. Both developing and mature neurons express major histocompatibility complex, or MHC, class 1 immune molecules throughout their lifespan. The brain was long thought to be devoid of these immune molecules, but their expression in neurons operate under a specific regulatory logic. They are directly controlled by the neuron's own electrical activity. This animated diagram shows the postsynaptic partner for these molecules. The activity-regulated MHC class 1 molecules on one side of the cleft bind directly to the D1-D2 immunoglobulin domains of the PIR-B receptor located on the dendritic spine. By acting as an inhibitory immune checkpoint, PIR-B enforces a strict ceiling on adult synaptic plasticity. It continuously restrains how much a cortical circuit is allowed to change. We know this because of the genetic knockout data. If you delete PIR-B in an animal model, the critical periods for visual plasticity never close at all. At the circuit level, removing this break leaves the animal with systematically higher spine density and more functional synapses. Their neurons exhibit enhanced long-term potentiation, making it easier to strengthen connections, paired with a distinct deficiency in long-term depression, making it much harder to weaken them. The brain evolved this immune-derived restraint system for a reason. By holding plasticity down, the PIR-B receptor maintains circuit stability, providing the physiological headroom necessary to consolidate long-term learning. Knowing that PIR-B regulates plasticity at the network level forces us to ask how it physically accomplishes this inside a single dendritic spine. Looking at the intracellular space of the spine, we can see the biochemical trigger. When PIR-B is engaged on the outside of the cell, its intracellular inhibitory motifs recruit SHP1 and SHP2 phosphatases. These phosphatases move through the cytoplasm to seek out a highly specific target, an actin-regulating protein called cofulin. The phosphatases activate cofulin by stripping a phosphate group from a single residue known as serine 3. At low physiological doses, this dephosphorylated cofulin performs a routine maintenance task. It neatly severs actin filaments, allowing the spine's internal skeleton to remain flexible and turn over normally. But if PIR-B is overengaged, the concentration of active cofulin pushes past the physiological range. At high doses, cofulin inverts its behavior. Instead of severing the actin filaments, it saturates them, bundling the mesh into rigid, insoluble one-to-one lattices. These rigid actin-cofulin rods mechanically occlude the narrow neck of the dendritic spine. Cargo cannot pass. Receptors and mitochondria can no longer be delivered. The spine is physically disconnected from the dendrite, and it collapses. Interestingly, a separate developmental glycoprotein called relin acts on this exact same serine 3 residue, but relin adds a phosphate group to stabilize the actin network, directly antagonizing the PIRB pathway. Two separate receptors, writing opposite structural instructions to the exact same molecular address. Alzheimer's synapse loss is the execution of this precise, mechanically occlusive structural program. The question is, what presses the button? To find the trigger, we have to return to the extracellular space and identify the pathological ligands hijacking this receptor in Alzheimer's disease. This diagram shows the extracellular mechanism. The disease state does not invent a new binding site. Pathological molecules deliver their instructions by slotting into the exact same D1, D2 domain on pure B that the healthy MHC class 1 molecules use. In 2013, the Shatz laboratory published a high-profile claim identifying the first purported pathological ligand. They reported that pure B is a high-affinity receptor for soluble amyloid beta oligomers. However, this data visualization tracks a rigorous 2019 standardized study by Smith and colleagues that directly challenged this. While synthetic amyloid bound the receptor in lab conditions, soluble amyloid oligomers extracted directly from human Alzheimer's brains registered zero detectable affinity for LILRB2. Because no independent laboratory has successfully replicated the binding with human-derived amyloid, the claim that amyloid beta is the primary driver of this specific receptor pathway must be carried as unresolved. But the mechanism itself did not fail. In 2025, the laboratory reported a second pathological ligand for LILRB2, the classical complement fragment C4d. C4d is generated when the immune complement cascade fires. Unlike other complement proteins that float away, C4d acts as a durable footprint. It covalently tethers itself to the exact surface where the cascade activated, binding to LILRB2 with extreme nanomolar affinity. The human anatomy data confirms its presence. Array tomography shows that C4d sits on roughly 30% of all excitatory synapses in the human cortex, and the protein is elevated fourfold in Alzheimer's patients compared to age-matched controls. While the amyloid interaction remains contested, the pure B receptor's capacity to structurally dismantle a spine is undisputed, and C4d provides a highly validated, anatomically confirmed trigger for that collapse. The discovery of C4d forces a correction to a major assumption in neuroimmunology regarding how complement proteins actually destroy synapses. On the left side of this diagram, you see the standard glial arm of synapse loss. The prevailing model dictates that complement proteins like C1q and C3 act as chemical tags. These tags mandate a microglia to arrive, attach via its CR3 receptors, and physically engulf the synapse. The right side shows the 2025 C4d finding. By binding to pure B, C4d establishes a parallel, entirely neuron-intrinsic arm of elimination. It triggers the internal cofalin collapse without requiring a microglial cell at any step of the process. A 2025 experiment by Brott and colleagues tested this causally. They used an osmotic mini-pump to infuse recombinant C4d directly into the cortex of adult mice over 4 days. In a wild-type brain containing absolutely no amyloid, no tau, and no background inflammation, the C4d infusion alone was sufficient to strip dendritic spines from the neurons. This bar chart tracks the necessity of the receptor. When the exact same C4d infusion was applied to a mouse bred without the PIRB receptor, the spine loss was entirely abolished. The dendritic density remained at baseline. Synapse elimination in Alzheimer's disease does not strictly require an external phagocyte arriving to eat the connection. A neuron can be commanded to dismantle itself. This topological map of the cortex highlights a recent development that changes the therapeutic arithmetic. LILRB2 receptor maps to two distinct addresses in the Alzheimer's brain, the pyramidal neuron and the microglial cell. When LILRB2 is engaged on microglia, it operates with the same inhibitory logic. It actively suppresses TREM2 signaling, acting as a brake that prevents the microglia from migrating and phagocytosing debris. This presents a rare therapeutic target. A single systemic antagonist against LILRB2 could theoretically act at two places at once, blocking the disassembly instruction on the neuron to protect the spine, while simultaneously releasing the brakes on the microglial cell to license clearance. The precedent for this intervention already exists in the clinic. Antagonist antibodies designed to block human LILR-B2, such as JTX-8064, have already cleared phase 1 and 2 trials in oncology, where the receptor acts as an immune checkpoint on macrophages. But an oncology program is not a neurology program. Those antibodies target peripheral myeloid cells. To act on synapses, they would require dedicated transport strategies, like transferrin shuttles, to achieve meaningful exposure across the blood-brain barrier. The larger issue is not pharmacological access. It is the physiological danger inherent to the mechanism itself. We must remember the biological prior. LILR-B2 is an evolutionary brake. The brain actively constructs and maintains this restraint system to stabilize neural circuits. If you systematically block this receptor in an aging human, you create a cortex that cannot weaken a synapse. Long-term depression is not a defect. It is half of the mechanism by which a circuit stores anything. A brain that cannot depress its synapses loses the capacity to consolidate memory and suffers severe interference between tasks. Before we can utilize LILB2 antagonists to halt structural erosion in Alzheimer's patients, the field must rigorously measure the lifetime cost of living with an unclosable critical period. Targeting this pathway does not repair a broken system. It actively chooses disinhibition over erosion. Whether the aging brain can tolerate that trade remains an unmeasured therapeutic window.